coming here in the fall, and she's joined my lab and is as a postdoc working on high-altitude adaptation in mice. But her prior work has all been on birds and looking at high-altitude adaptation in siskins. So she's a person who's interested in questions and is not taxonomically bound to one group, but is interested in all kinds of things, including, of course, the fascinating mice, in addition to the siskins. The fun fact about Libby is she writes that the best birthday present I ever received was getting to prepare a female cock of the rock, the national bird of Peru, on my birthday in northern Peru. So that was a pretty good birthday present. And it says a lot about you, right? So we're delighted to have her here as a postdoc, and today she'll tell us more about high-altitude adaptation in siskins.
Thanks for coming. Today I'm going to talk to you about this amazing array of finches and how they inform us about important evolutionary processes like dispersal, diversification, introgression, and adaptation in a biodiversity hotspot, the Andes of South America. Before I get into the subject of my talk, I'd like to acknowledge some of the people who have supported my research along the way. The work I'll present today was done during my dissertation with Chris Witt at the University of New Mexico, and it depended heavily on fieldwork conducted by students and professionals at the Museum of Southwestern Biology, some of whom may be in the audience, like Fred Benham and Ashley Smiley. Collecting at high elevation in the Andes is not easy. So here you see us exiting a site at high elevation in Peru, where we're hand carrying our specimens out and our equipment is tied to donkeys. I'm extremely grateful to everyone who contributed to these challenging field expeditions. I'd also like to acknowledge the many other prescient collectors who acquired samples from across South America, as well as the museums who donated tissue and specimen access for this work, including the MBZ. My research interests focus on three central themes and the interplay between them. First, diversification history, by which I mean large-scale patterns of speciation and extinction, and how diversification patterns are the result of both stochastic evolutionary processes and idiosyncratic history. Second, I'm interested in interspecific introgression, which is the exchange and retention of genes between independent lineages like species. It's becoming clear that introgression is a common phenomenon across the tree of life, and its pervasive presence impacts both our attempts to reveal evolutionary history through phylogenetics and demographic modeling, as well as our understanding about the movement of adaptive alleles across a landscape. Third, I am interested in understanding adaptation, the predictable nature of adaptation to known selective challenges like high elevation, and the genetic sources of beneficial and deleterious mutations. These three concepts do not exist in isolation from one another, and one may hold essential explanatory power for understanding another. Today I will discuss these three concepts by considering the evolutionary history of a young avian radiation South American siskins that span an extreme environmental gradient. This is an outline of my talk. First, I'll introduce the study system of the South American siskins, and then I will tell you three short stories about their evolutionary history. I will ask how dispersal and vicariance have contributed to biogeographic patterns in and outside the Andes. To do this, I will describe the spatial and temporal patterns of diversification in the siskins and goldfinches of the New World. Second, I'm going to test for the presence of interspecific introgression across the genome among sympatric species despite phylogenetic uncertainty to elucidate the role of introgression in rapid radiations. Last, I'm going to address the role of natural selection in generating intraspecific diversification and providing opportunities for introgression in a key target of selection across elevation, the oxygen transport protein hemoglobin. I hope this deep dive into the evolutionary history of siskins will help you see insights into the evolutionary processes that generate the wonderful biodiversity around us. We'll begin with the siskins. The South American siskins are a rapid radiation consisting of 11 currently recognized species found across South America. The clade includes species that are distributed across a wide range of elevations, including the hooded siskin, pictured here, that can be found from sea level on the coast of Peru up to 5,000 meters in elevation. The clade also includes species that are tightly restricted to a narrow elevational zone, like the black siskin, which is confined to above 3,500 meters, and mid-elevation species like the olivaceous siskin. The Spinous siskins reach their highest diversity in the high Peruvian Andes, where 3 species co-occur during the breeding season and a 4th joins them during the non-breeding season. These 2 images give you a basic idea of the morphological variation and geographic extent of the South American siskins and their close relatives, the goldfinches in North America, as well as siskins in North America, Central America, and Eurasia. The South American siskins are indicated in the photo by black asterisks, and the geographic ranges of all New World siskins and goldfinches are depicted in different colors in the panel on the left. You can see that much of the diversification within Spinous follows the spine of the Andes, which stretch from north to south along the western coast of South America. The extensive sympatry of siskin species and the variation in the elevational ranges of Andean species makes this radiation well suited to address diversification, introgression, and adaptation in the context of a chronic abiotic stressor, high elevation. High elevation environments pose unique challenges to organisms that colonize, diversify, and persist in them. High elevation environments are characterized by greater seasonality over the year and greater diel variation in temperature over the day, colder temperatures, and reduced partial pressure to oxygen, or hypoxia. This latter condition, the reduction in available atmospheric oxygen, is of particular concern to metabolically active animals like birds and mammals, which both regulate their internal temperatures through metabolism and, in the case of birds, can increase their energy consumption 15-fold during flight. High elevation environments also occur across the world. Thus, the study of them can shed light on the predictable nature of evolution to a similar abiotic selective pressure like hypoxia in independent lineages. The morphological diversity, elevational range variety, and the recent diversification of Spinous gives us an opportunity to examine the process of speciation and extinction in the Andes, a region of exceptional avian biodiversity and endemism. To better understand the role of high elevation environments in diversification, Chris Witt and I used a time-calibrated phylogeny and biogeographic and sample reconstruction to characterize how siskin species diversified into and out of the Andes. Dated molecular phylogenies from birds and other taxa support the hypothesis that Andean biodiversity is primarily the result of allopatric speciation facilitated by orogenesis and climatological shifts since the mid-Neogene. Several meta-analyses of neotropical bird diversification patterns have suggested that avian lineages experience different tempos of diversification in the Andes and the adjacent lowlands. Andean bird species tend to have speciated more recently than lowland species, and lowland bird clades typically show a significant deceleration in their diversification rate, while most Andean clades do not. The ultimate reason for this pattern could be regional disparities in the opportunity for vicariance. High elevation taxa tend to have linear geographic ranges that may be prone to fragmentation. Alternatively, lowland species are more likely to be impacted by marine incursions due to rising sea levels associated with climate variation. Climatic cycles during the Pleistocene comprise one of the principal vicariant causes of bird diversification in South America. The most thorough examination of this phenomena, the Andean and non-Andean taxa differ in their diversification patterns, was conducted by Jason Weir. He found the high average diversification rate of Andean taxa was driven by the inclusion of 3 recent rapid radiations, including the Spinous siskins. However, the Spinous phylogeny that he used had poor geographic representation and low resolution, which makes the result of this meta-analysis difficult to interpret. With the aim of improving the phylogenetic hypothesis of Spinus through increased geographic and taxonomic sampling, we asked, was diversification faster in Andean Siskin lineages relative to non-Andean ones? And did Siskin lineages move into and out of the Andes at different rates? Our objectives were to develop a time-calibrated phylogeny of New World Siskins and goldfinches, and calculate region-specific diversification rates and interregional dispersal rates between the Andes and adjacent lowlands. To meet these objectives, we sequenced all the New World Siskins and goldfinches except one South American species we couldn't obtain, and included as many as 5 geographic populations for widespread species like the hooded siskin, colored yellow in this figure. The colored dots indicate sampling localities that we included. We sequenced all individuals for 3 mitochondrial genes and 2 nuclear introns. We used star bees and mitochondrial gene-specific substitution rates to estimate a time-calibrated phylogeny in a coalescent framework. We found the South American siskins form a monophyletic clade labeled on the figure, with a most recent common ancestor just over 0.5 million years ago, dating to the late Pleistocene. We also recovered strong support for a central Andean clade that includes 5 species that occur at mid to high elevation in the Andes of Ecuador, Bolivia, and Peru. I'll also point out that the widespread hooded siskin, Spinus magellanicus, appears in 2 separate places in this tree, with birds from northern Argentina falling outside the central clade. We used Bayesian binary MCMC and S-DIVA to reconstruct the ancestral range of the New World siskins and goldfinches. Importantly, the ancestral state reconstruction, as well as the diversification rate analyses I'll talk about next, explicitly incorporated phylogenetic uncertainty by using an additional 500 alternative trees that were drawn from the posterior distribution produced in Star Beast. We found that siskins colonized the New World from the north in a stepping stone fashion, from North America into Central America, and later into South America and the Andes. We recovered an exceptional diversification rate for goldfinches and siskins in the New World throughout the Pleistocene at about 1 species per million years. For perspective, this is more than 4 times higher than 75% of all bird diversification rates that have been previously reported. We also found the best diversification model was a pure birth-death model with no indication of diversification decelerating over time. Comparing Andean and non-Andean diversification models, we found the best model using geographic SSE was a model using 2 rates of speciation and dispersal between the 2 regions. In this model, the speciation rate in the Andes was 2.5 times greater than the non-Andean species, and the dispersal rate from the Andes into the adjacent lowlands was over 3-fold that of non-Andean residents. We successfully constructed a time-calibrated tree including all New World goldfinches and siskins, and found that the siskins in South America represent a single radiation. Our analyses revealed an exceptional diversification rate across the New World clade and especially high rate of diversification in the Andes. This result supports one of the major macroevolutionary hypotheses on bird diversification in South America, that Andean bird lineages overall exhibit rapid and ongoing diversification in the Andes, driven in part by climatic variation during the late Pleistocene. Second, our finding that Andean Spinus have a high dispersal rate into adjacent lowlands supports the hypothesis that the Andes serve as a species pump, contributing a disproportionate number of lineages to lowland regions. The central role of the Spinus siskins to Weir's 2006 meta-analysis and our confirmation of their exceptional diversification rate also suggests that there may be something special about the diversification potential of these finches. The dispersal propensities of siskins, including eruptive migration, may predispose these finches to successfully colonize new regions. Trevor Price and others hypothesized that eruptive behavior, specifically the nomadic and unpredictable migration of finch flocks in response to food shortages, may be one of the shared traits that have led to successful radiations such as the Hawaiian honeycreepers and crossbills, as well as other modern seed-eating birds such as Darwin's finches and the cappuccino seed-eaters. Through our multi-locus analysis, we learned about the pivotal role of the Andes in spinous diversification. We also found an unusual pattern where, within this radiation, species that are sympatric in the high central Andes were also each other's closest relatives. In fact, certain species with extensive sympatry also shared common mitochondrial haplotypes. This striking pattern could be the result of interspecific introgression among Andean spinous, and we set out to investigate this possibility. Alternatively, this pattern could be the result of incomplete lineage sorting. Understanding the contribution of both these factors on phylogenomic analyses is an important current topic in evolutionary biology, as genomic data sets become more common and frequently reveal discordant gene histories within the genome. Across the tree of life, it's becoming clear that there are certain evolutionary relationships that are persistently challenging to resolve at both deep and shallow timescales. In the network diagram of Darwin's finches, this uncertainty is represented by the dense cluster of connected lines at the center that represent alternative pathways to connect the basal nodes of the evolutionary tree. An important component of this uncertainty is incomplete lineage sorting, where ancestral polymorphism is retained within populations across multiple divergence events. The probability of incomplete lineage sorting is increased in rapid radiations, where diversification events occur in quick succession. However, another source of phylogenetic uncertainty is introgression. Introgression is the movement of alleles from one lineage into the gene pool of another. Recent genomic studies across a diverse set of taxa, including the Darwin's finches, have shown that introgression is likely more common than previously thought, and it can have a significant impact on our ability to resolve evolutionary relationships. The presence of both phenomena in rapid radiations presents something of a conundrum, since rampant integration could result in a phylogeny that is impossible to resolve. This creates a paradox because robust tests for integration require the phylogeny to be known without error. For this study, we identified two crucial questions. First, how common is introgression in rapid radiations? Second, how does introgression in rapid radiations impact our ability to study these clades? To these ends, our specific objectives were to extend the taxonomic and geographic scope of the study of introgression in rapid radiations, and to develop an approach in which introgression can be assessed despite phylogenetic uncertainty. To achieve these objectives, we focus in on the Andes. This is a picture taken at 3,700 meters in the Peruvian Andes. Here we can find 4 species of closely related siskins co-occurring during the non-breeding season and 3 during the breeding season. Here you can see the geographic overlap of these 4 species: Spinus crassirostris, Spinus atratus, S. europigialis, the widespread S. magellanicus. I'll be using this color scheme to indicate species for the remainder of this section. The immediate proximity of closely related species in the high Andes may challenge species limits and make this a good system in which to test for the presence of interspecific introgression in a rapid radiation. Our null hypothesis for this study was that introgression has not occurred among spinous populations. Our alternative hypotheses were that introgression has occurred in the nuclear genome of Andean spinous, and that mitochondrial haplotype sharing reflects interspecific introgression events among Andean spinous. We tested these hypotheses by conducting genotyping by sequencing, a DD-RAD-like approach on our 4 focal taxa and 1 outward S. cucullatus that's found in northern South America. For GBS, samples for the widespread S. magellanicus were taken from 3 different regions. The localities are indicated as large dots with the northern population indicated in yellow, Bolivian populations in pink, and lowland Argentina birds in orange. We also sequenced 3 mitochondrial genes from S. magellanicus from all the localities with the dot. We analyzed the population structure of our nuclear and mitochondrial DNA with phylogenetics, admixture, a clustering analysis similar to structure, principal component analyses, and haplotype networks. We also performed formal introgression tests using several tree-based approaches. These methods explicitly incorporate incomplete lineage sorting. After processing, we recovered about 45,000 SNPs from our GBS sequencing. However, we varied the amount of missing data for different phylogenetic methods. I won't discuss how we handled missing data here, but I'm happy to talk about it if you have questions later. We found a robust phylogenetic hypothesis using maximum likelihood. In this phylogeny, S. magellanicus comes out in two places in the tree. There is a northern clade from Peru, here, and a southern clade from Bolivia and Argentina. We also find S. atratus is sister to the northern S. magellanicus clade. However, when we ran SNAP, we recovered 2 additional topologically distinct trees. When we recovered a fourth conflicting topology using SVD quartets. Note, in all of these trees, S. magellanicus is not monophyletic, and the sister relationships of S. atratus and northern S. magellanicus is present. Topological differences arise from uncertainty in the placement of the root. Here, the within clade relationships are identical between these 2 trees. However, the placement of the outgroup is different. Topological differences are also the result of conflicting relationships between the clade with S. atratus and northern S. magellanicus, and the 2 lineages S. crassirostris and southern S. magellanicus. In the lower trees, the southern S. magellanicus, in orange and pink, are sister to the S. atratus northern S. magellanicus clade in black and yellow, as indicated by the red arrows. This relationship is not present in the other 2 trees, as indicated by the black arrows. When we analyzed our GBS data using principal component analyses, we found S. crassirostris and S. europegialis on different axes. When we removed these species to investigate the variation within the remaining species, we found 2 interesting patterns. First, S. magellanicus from Bolivia is equidistant from S. atratus and S. magellanicus from Argentina. This result could imply shared ancestry for the Bolivian population, though the pattern is also concordant with other hypotheses. Second, S. magellanicus from southern Peru were closer to S. atratus on the PC2 axis than they were to central Peru S. magellanicus. In our clustering analysis with admixture, we found several interesting results. First, the program was unable to distinguish between northern S. magellanicus and S. atratus. Second, some of the S. atratus individuals appear to have high proportions of shared ancestry with the southern S. magellanicus. Last, some northern S. magellanicus individuals appear to have shared ancestry with S. crassirostris. PCA and admixture results together provide evidence for introgression among species, but we formally tested these using a tree-based introgression test. To test pairwise introgression in light of our phylogenetic uncertainty, we designed tree-based tests that relied on regions of strong support that were recovered across different topologies. To briefly illustrate how these tree-based tests work, consider if we're trying to test for introgression between taxon 2 and 1, given this phylogeny. We can draw a 4-taxon phylogeny to depict the relationship between taxon 2 and 3. We can then test if our genetic data fit this tree proposed. If our data don't fit, this will be evidence for introgression. As I go through the results, I will point out if a test provided evidence for introgression based on the Raxamel tree as well as other trees. Further, since all methods have different assumptions, we consider the results of our population structure and tree-based tests together. Best support for introgression will be if introgression is supported across trees or in multiple methods. Evidence for introgression between S. crassirostris and northern S. magellanicus comes from tests based on the Raxamel tree, all other trees, and admixture. There is evidence for introgression between S. petratus and S. magellanicus from southern Peru from formal tests and admixture. PCA result was also concordant with introgression. Evidence for S. atratus and Bolivian S. magellanicus introgression was supported across methods, but not all phylogenies. Introgression for S. europigialis and northern S. magellanicus was supported in multiple formal tests pertaining to the Raxamel topology, but no other trees, and there is no evidence of introgression from the other methods. With the GBS data, we identified strong support for certain evolutionary relationships across phylogenetic methods. We used the regions of strong support, and considering phylogenetic uncertainty, we were able to identify putative pairwise introgression patterns. By combining population structure analyses with formal introgression tests, we report strong evidence for 3 to 4 introgression events among closely related sympatric spinous impacting the nuclear genome. In the mitochondrial genome, we found extensive haplotype sharing among certain species. I want to point out that S. magellanicus is found in 3 parts of this network, sharing haplotypes with S. crassirostris at the top, sharing haplotypes with S. atratus at the bottom left, and lastly, S. magellanicus from Argentina form their own distinct haplotype group in the lower right. When we plot these major haplotype groups found in S. magellanicus across geography, we see that the haplotype distribution is clearly spatially structured. Further, though the GBS data supported a close relationship between S. magellanicus from Bolivia and Argentina, the mitochondrial DNA shows Bolivian birds from mid-elevation are fixed for the S. atratus haplotype. The mitochondrial DNA haplotype sharing patterns mimic 3 of the nuclear DNA introgression events we identified. From this study, we conclude that strongly supported conflicting topologies for the spinous tree can be attributed to a history of introgression. Second, the use of multiple methods is a powerful approach for revealing subtle evolutionary history from DD-RAD-like data. And third, the spinous diversification history supports a more reticulate tree of life. The last story that I'll talk about today is how high elevation adaptation has impacted the evolution of spinous. We found earlier a close relationship between elevation, dispersal, and diversification. Here we look at the role of natural selection in generating intra-specific diversification and opportunities for introgression in a key target of selection across elevation, the oxygen transport protein hemoglobin. In our results, I will describe a striking pattern in the functional variation of hemoglobin across the spinous clade that is consistent with adaptive introgression, as well as evidence for local adaptation to high elevation within a widespread species. The tetrameric protein hemoglobin is responsible for circulating oxygen through the body and is a critical component in adaptation to elevation across the avian tree. For example, in this comparison of sister pairs of Andean birds, Natarajan reported that birds residing at high elevation exhibited a stronger binding affinity between hemoglobin and oxygen in both adult hemoglobin isoforms. This pattern is shown in this figure by the majority of dots that are located below the central line, indicating that high-altitude species tend to have a lower P50 and thus stronger binding affinities than their low-elevation counterparts. This result suggests that hemoglobin may be an initial target for local adaptation in populations distributed across a wide elevational range. However, local adaptation may be easily eroded through gene flow. Understanding how hypoxia-associated natural selection impacts populations of siskins distributed across an elevational gradient can help us understand if it is an initial component in the process of diversification in siskins. Hemoglobin is also a probable target for adaptive introgression, since hybridization can introduce an adaptive allele into a population at a higher frequency than a de novo mutation will arise, and previous work has shown that highly adapted avian hemoglobin loci often are the result of multiple mutations. Interestingly, the order in which beneficial mutations accumulate in hemoglobin can dictate whether or not the optimal hemoglobin haplotype is ever reached. So in this image, you can see the oxygen binding affinity of a high elevation population on the right and a low elevation population on the left with a lower binding affinity. In between, you see the binding affinities of mutations accumulated from right to left in a unique order, with only some of those trajectories consistently performing better than the original given a lowland population. This property of mutation order dependence in hemoglobin means that highly adapted haplotypes may take a long time to produce through the random accumulation of de novo mutations. Alternatively, the introduction of a novel allele from another population with multiple synergistic substitutions may produce a large selective advantage immediately. At the same time, the performance of hemoglobin alleles can also be intrinsically linked to the genetic background of a population, meaning functional improvements on performance may only be realized on the background of the original population. Hemoglobin is coded by two gene complexes in the avian genome. You can see here the components of two protein isoforms that contribute to adult hemoglobin. HBD includes two subunits of alpha D and two of beta A. HBA includes two subunits each of alpha A and beta A. The genes alpha pi, beta H, beta rho, and beta epsilon contribute exclusively to embryonic hemoglobin. To develop a complete perspective on the role of hemoglobin in spinous high elevation adaptation, I made target capture probes to sequence the complete coding regions for all 7 hemoglobin loci. I asked, how has introgression impacted the distribution of functional hemoglobin alleles? And how has adaptation to high altitude hypoxia promoted local adaptation within a widespread species? My objectives were to test the association between functional hemoglobin alleles and elevational range across the spinous clade, and to test if non-synonymous mutations varied significantly across elevation within the widespread Peruvian S. magellanicus. We sampled 9 out of 11 South American species and 1 outgroup, S. notatus. For S. magellanicus, we sampled across 2 elevational transects, 1 in Lima and 1 in Ancash, with about 40 birds per transect. And we used 150 base paired end sequencing with a target capture approach to sequence all 7 hemoglobin genes, as well as 450 exonic regions distributed across the genome. For the remainder of the talk, I'm going to focus on non-synonymous mutations that we found in the 7 hemoglobin. As we consider the clade-wide results, I'll first remind you of what we found in the previous work. Recall that Bolivian hooded siskins were closely related to lowland Argentinian hooded siskins in all the previous phylogenetic analyses. If the hemoglobin loci reflect phylogenetic history, hemoglobin variants should be shared between mid-elevation Bolivia and Argentina. Alternatively, incomplete lineage sorting and de novo mutations may produce an alternative pattern with no clear reference to the phylogeny. Last, adaptive introgression could be would be indicated by a strict tie between non-synonymous mutations and elevation. For now, I'm just going to focus on the patterns that I found. Calculating their probability in order to assign the source of the pattern is in progress, and I'm happy to talk about it with you after the talk. Looking at the loci, which code for adult hemoglobin isoforms, I found 2 non-synonymous mutations in hemoglobin alpha A that are strongly associated with elevation. At alpha A34 here, there is a strict correlation between elevational range and the possession of the ancestral amino acid alanine or the derived amino acid threonine. This pattern extends to the high elevation Bolivia lineage, which possesses the derived allele. Alpha A34 is a site that has been previously shown to provide hemoglobin binding affinity shifts in nightjars. In a gene tree, including the entire locus, which had some flanking and intronic regions, there is strong support for this bifurcation, with bootstrap support over 90. At alpha A120, mid-elevation Bolivia again shares a derived allele that is found only in high-elevation Spinus populations. At hemoglobin alpha D, there are 2 sites, alpha D10 and alpha D120, where high Bolivia again shares 2 derived alleles found only in high-elevation species, with a nearly strict association of high elevation and the amino acid glutamate at alpha D120. Again, the species that occupy elevations below 2,000 meters possess a single exonic haplotype. Last, in hemoglobin beta A, the only beta component in adult avian hemoglobin, there is 1 shared derived allele found in high-elevation Bolivian birds and high-elevation Spinus species, though the ancestral allele alanine is still present in both Bolivian and some high-elevation species. In this case, we have experimental evidence through collaboration with J. Storrs at University of Nebraska-Lincoln that the derived allele threonine at beta A21 increases the hemoglobin oxygen binding affinity under certain conditions over the ancestral allele. Thus, across the Spinus clade, there are derived non-synonymous mutations in each of the coding adult hemoglobin loci, which are closely associated with high elevation, including the Bolivian hooded siskins at 2,500 meters. In each of these cases, the Bolivian population possesses the alternative allele, while the Argentinian population possesses the ancestral lowland allele. 2 of these non-synonymous mutations are associated with a functional difference in hemoglobin binding affinity, and in the case of beta A21, there is direct experimental evidence of binding differences for the high and low mutations. In light of the multiple introgression events I identified earlier, including one between the high elevation specialist, the black siskin, and the high elevation Bolivian hooded siskin, these results suggest adaptive introgression of high elevation adapted hemoglobin loci into the Bolivian hooded siskin. Adaptive introgression is rare in vertebrates. However, in one of the best recognized cases, an allele from an archaic human lineage, the Denisovan, facilitated adaptation of modern humans to a high elevation environment, the Tibetan Plateau. This result in siskins confirms that introgression from close relatives may be an important source of genetic variation for high elevation adaptation. Last, I will summarize the differences of the 7 hemoglobin loci we found across 2 elevational transects on the west slope of Peru in the hooded siskin. I'll just highlight the most interesting non-synonymous SNPs from here. First, we found a linear but not statistically significant trend in allele frequency change associated with high elevation in hemoglobin beta 21A. The derived functionally significant allele threonine is present at higher frequencies in the high elevation populations. We also found a statistically significant trend between allele frequency and high elevation for alpha 60 pi in Ancash and beta 135H on the Lima transect. We also used FST outliers between the high and low populations on both transects to identify SNPs with significant differentiation along each transect. And for comparison, we included SNPs from the 450 exonic regions we sequenced. We found that beta A21 was a significant outlier on the Ancash transect, and the SNP at alpha pi 60 was a significant outlier on the Ancash transect and nearly on the lemma. Last, beta H135 was a significant outlier on both transects. We consider that having multiple lines of evidence for significant associations between allele frequency change and elevation to be strong candidates for hypoxia-related natural selection in the Peruvian hooded siskin. Summarizing across these analyses, we found moderate evidence for differentiation for beta 21A in Ancash, as well as on both transects, alpha 60 pi and beta H135. This is the first time that embryonic hemoglobin, so alpha pi and beta H, has been found to be under natural selection associated with elevation, and it deserves a little attention because it is a fundamentally different problem than the well-described challenge of providing oxygen to a fetus during pregnancy in a hypoxic environment. But it becomes clear how important having the right hemoglobin binding affinity is in the embryo when we consider the diffusion pathway which oxygen travels to get from the air into the avian embryo in the early stages of development. Here, you can see the oxygen diffusion pathway is limited at multiple steps, including the slow diffusion of oxygen from a gas across the aqueous inner membrane of the shell, as well as traveling across the egg albumin and diffusing through the yolk sac into the embryo. Thus, in the early embryo, the oxygen is extremely limited, and the restriction of oxygen controls the rate of growth for the embryo. By the end of a week in a chicken egg, this diffusion pathway has fundamentally changed to allow more oxygen to reach the embryo. These changes include the introduction of an air sac within the respiring egg, which you can see it in the top right figure, as well as the movement of the yolk sac to be adjacent to this airspace. The yolk sac also becomes vascularized, enabling it to serve as the principal organ for gas exchange for the embryo. The task for the embryonic hemoglobin loci alpha pi and beta H are unique within the development of the egg, and here we have shown that they are under selection across an extreme gradient in atmospheric oxygen. Since these rate-limiting conditions in the early embryo are probably shared across avian diversity, I suspect embryonic hemoglobin is under selection with respect to hypoxia in many high-elevation taxa. We just haven't looked for it yet. So in conclusion, we found that beta 21A is a novel biologically relevant mutation for interspecific and intraspecific adaptation to high elevation. We report that alpha pi 60 and beta H135 are SNPs that appear to be under selection across both elevational transects in the Peruvian hooded siskin. These loci are expressed exclusively in the avian embryo. Last, despite the high dispersal propensity of finches, allele frequency differences in hemoglobin have accumulated over less than 80 kilometers as the finch flies. So, today I've told you 3 interlocking stories about diversification in the South American siskins. We went from continental diversification patterns and the significance of the Andes in siskin speciation to the role of introgression and how it's impacted sympatric Andean siskins, as well as the challenge for testing for introgression when phylogenetic uncertainty persists. And we ended on the outsized role that adult and embryonic hemoglobin play in the evolutionary history of birds, including the South American siskins. I hope I've convinced you that a deep look into the evolutionary history of a single clade can reveal important insights into the evolutionary processes that shape the biodiversity around us. And I hope that if this work interests you, you will stop me in the hallway or chat with me at coffee, and I'm happy to talk about the future that I see with siskins. I'll thank again the people who have helped me with this work and my funding sources. And with that, I will take questions.
My question is, so you found evidence of introgression through your discordance between your Avian and Raxamel trees. My question is, when you're looking at hemoglobin, how do you know these point mutations are the result of an introgressed tract or from convergence?
Yeah, so I mean, one of the things that is pretty convincing is that this is a very young radiation, and so these derived alleles, we don't expect there to have been enough time for the same allele to have arisen in multiple lineages over this really short period of time. So that's one of the reasons. And then, yeah, there are some issues with deciding exactly whether or not it's adaptive introgression, and that has to do with the uncertainty in the phylogenetic tree. And so that's something that I'm currently struggling with, but I think I have a solution. So.
Do you see synonymous mutations that are synonymous rather than non-synonymous that are common to the introgressing parent species?
Yes,
Okay.
and so there are some some synonymous mutations that inform, but there are actually very few synonymous mutations that we found in the coding regions, and that's what we have the best coverage for.
So Libby, you targeted hemoglobin, right? So you don't have flanking lesions to look at the decay of, to look at the length of the introgress segments. Right?
No, that would be great.
That would help you date the age of those alleles.
Right, yeah, so, I mean, we just didn't catch enough of that for it to be powerful. Yeah.
So at the embryonic stage, the birds are essentially ectothermic, right? They're not generating their own heat.
Yeah.
So do we have any data on what egg temperatures actually are along elevational gradients?
Oh, that's a great question.
And then I'm sort of asking because basically if the female is, or do both parents sit on top of their eggs?
It depends. I'm not actually sure about in
Okay.
Siskins, but I mean.
But I'm just curious if the adults are maintaining the eggs at the same temperature at different altitudes, that would be a pretty strong selective pressure for the differences in hemoglobin. Whereas if there are differences in temperature, you might not expect this because they would have a lower metabolic rate through the cooler climates.
Yeah, I mean, I do think probably that the birds at high elevation are trying to maintain the same, are behaving the same way, sitting on their nest for an amount of time. But it's a good point. I guess you would need eye buttons and find a bunch of Siskin nests.
Make some fake eggs,
Yeah. Yeah.
You mentioned that one of your species doesn't breed at the high elevations. Is that one missing the embryonic mutations on the hemoglobin genes?
Oh, it does breed at high elevations. It's just not sympatric in the Peruvian Andes.
Okay.
But it does breed at high elevations and only at high elevations. And then it only, we think, our natural history understanding of these birds isn't perfect, but it seems to only be present in the Peruvian Andes during the non-breeding season, what we think is the non-breeding season. Yeah.
I may have missed it, but do you have any evidence for inferring the date for introgression events? And is there any possibility it's still going on?
I think it's, so if you think about the admixture results, there is some of those results have really high proportions of shared ancestry between two lineages, and so that kind of suggests that it might be an ongoing issue, and all of these birds are sympatric with each other now, and so yeah, that's a good question, and certainly something we'd like to do, but not something that we can do with GBS data. Yeah.
I just don't recall, does the mitochondrial introgression pattern match the hemoglobin introgression pattern? Do you think that might also be adaptive?
Yeah, it matches perfectly. So the Bolivian birds are fixed for the black Siskin mitochondrial DNA. And then the Bolivian birds also particularly for alpha A, have a perfect association, are fixed for the high elevation hemoglobin allele.
You think the mitochondria might also be under selection?
It seems very, very likely, but there aren't very many specimens from that area, or to dive into it, basically, you have to sample specimens from the 1980s.
Is the introgression direction the one you would expect? Is it a high
For
altitude
Bolivia, I would
mitochondrion
say so,
that's being
yes.
introgressed into the high elevation?
Yeah, so you have this, so the black Siskin is this high elevation specialist that appears, like, we know that there's evidence for introgression in the nuclear genome, and they have been the donor of the mitochondrial haplotype for the Bolivian birds, and those Bolivian birds have a low, have a close relationship with a low population, the Argentine population. So we suspect that they're secondarily colonizing the high Andes, and doing so, it's much easier if you have the right hemoglobin.
And perhaps the right
And
mitochondrion?
perhaps the right mitochondrion.
You know which chromosome the hemoglobin's on in these birds?
I do know, but I don't know off the top of my head. Though the alpha globins and beta globins are in different chromosomes.
It seems to me that you're making the assumption that you have all the players.
Mm-hmm.
But what if some of the players just went extinct?
Well,
If they were here yesterday, or you haven't said it.
you mean for introgression or?
Yeah, right.
Yeah, I mean, that is a possibility. I mean, it does suggest the fact that we see the introgression patterns closely mirroring current sympatry suggests that the introgression results that we have are reflecting introgression between those lineages rather than another source. Yeah.
Are the mitochondrial sequences identical?
Mm-hmm.
So it's recent.
Yep.
Yeah.
They're exactly
All right, very cool. Well, thank you very much.
Bring back fond memories.
You needed that.